So what I'm going to talk about is real-world experiences in operating a collaboratory, and in this case, it's something called the Protein Data Bank. So the PDB is a single international repository for all information about the structure of large biological molecules. And it's an archival database with hundreds of thousands of users who depend on the data, and that's not an exaggeration. So here's a picture of the kinds of things that are in the PDB. These are large biological molecules, and this picture is organized according to their biological function. So on the right, for example, are protein systems that are involved in structural things like collagen, which is the most abundant protein in your body. And then on the left are some of the early very small molecules like hemoglobin and myoglobin. And for example, this picture showing the DNA, a rope of DNA with the different kinds of proteins that bind to DNA. So the PDB is a repository for information about these molecules. So it contains, in addition to the coordinates, a potential of 3,000 metadata, but actually about 400 metadata are deposited per structure. And this shows the growth of the PDB from its inception. So we're talking now about a resource that is 35 years old, and I was there at the beginning. And so this is really more about sociology than about technology, okay, right now. And I can assure you that at each step along the way, we've had to embrace the new technologies while at the same time keeping the data sacred, and that has been a real issue. So a lot of the things that you talked about with respect to the science of science of science is really important. So you have to keep the science, keep the scientists happy as you march through the technologies. So it started in the 1970s as a grassroots movement to archive data. So in the 1960s, the first protein structure was solved in 1957, and in the 1960s, people began to think, gee, we'd like to have these data. And there was a meeting in Cold Spring Harbor in 1971 to bring the groups of people together who, at that meeting, there were the people who wanted to have data, and there were the people who were producing the data. And frankly, it was at the bar in Cold Spring Harbor that we convinced somebody, I don't know where the pointer is, but this person here, Walter Hamilton, was a scientist at Brookhaven National Laboratory, and he was really interested in this subject. And so he said he would do this, and that's how the PDB began, with 7 structures, and it wasn't until 1975 that the funding came. Now, to give you a sense of what it was like, to talk about people. So here are some of this is the car that took us to Cold Spring Harbor, where we rallied in the early 70s to make the PDB happen. And that Cujo was a very important car that allowed us to get around to convince people that this was an important thing that we had to do. And being that it was in the early 70s, we did everything by petitions and carrying on and stuff. And so on the left is a copy of one page of the petition that we wrote to I don't know who, that we needed to have this repository. Because we got somebody politically strong to be involved, he was able to do the right thing and make this international and get the paper published in Nature, and get this thing going. Now, on the right is something that probably one or two of you might know what that is. It's a punch card. That's a punch card, and that's not actually a Chad because the thing is pushed out. But anyhow, it's just a joke. But one of the things you need to know about what happened when the PDB began is we were using ARPANET to move data around. So what would happen is that I had a little teletype computer where I could type things saying, and I was in Philadelphia at the time, telling the people at Brookhaven to do things like mount a tape or grab the cards or do something. And that was our way of, that was our first use of what is now the internet. And this project depended on that. And it was already distributed between Brookhaven, Philadelphia, and Texas. In the 1980s, thank God, technology took off, and that it meant that molecular biology came of age where it was possible to make large amounts of protein. The instrumentation for collecting data there was a big leap forward where you could use synchrotrons at the National Lab to get intense sources. Computer hardware and software improved. And because of that, structural biology was able to focus on medical problems. And at the same time, because of the importance of these structures to medicine and to drug design, there was a big push to promote data sharing at a time that was not popular. So this was in the early 80s, and it was coincident with the beginning of the AIDS epidemic, and that it was related to why the data sharing was thought to be so important. And the International Union of Crystallography finally published guidelines for requiring data deposition into the PDB. So again, community push to make this happen was very, very important. In the 1990s, we had the number of structures begin to increase exponentially. The complexity of the structures increased. New kinds of databases began to emerge. There were more structures being determined by cryo-electron microscopy, which meant we got really big structures. There was the plan for structural genomics, which meant to determine the structures of all proteins, basically. And the user community for the PDB expanded dramatically. And so the NSF had a competition, an RFP. We formed an organization called the Research Collaboratory for Structural Bioinformatics, consisting of Rutgers, UCSD, SCSC, and NIST. And together, we said we would be able to manage these data and get the data through the pipeline rapidly and have very good curation. All of these things, I think, as you said, require people and people getting along and people being happy and all kinds of stuff like that. And that is really important because it's a drudgery. This whole thing of archiving data is very difficult and very stressful, and we're not allowed to miss a beat. So this is a party celebrating getting our grant. This picture at the bottom shows the leadership team from when we got the grant in 1998, and some of you might recognize the NSF. That's the Antarctica sort of exhibit they had. That's the tent right behind us. And then somebody made a cake for the celebration, and all these words mean something to us in terms of fix it with max it and all that. It has to do with our software. I'm not talking about any of that here. I'm just talking about how people have to just keep working together. So who does what? At Rutgers, we do data ins, and that means one of the big issues is standards. We developed, starting in the early 1990s, we were part of a team to develop a data representation for crystal structures, and we developed a dictionary of 3,000 terms that defined rigorously every single step in the experiment and the result, and it was put in a relational framework. It isn't some crazy language that we made up. It doesn't matter. The issue is that we have a very rigorous syntax, a very rigorous semantics. So when XML came along, we could just translate it. XML is not important. Arab language isn't important. What's important is that we had a very clear set of definitions. This thing of setting first for X-ray, we then got into NMR, cryo-electron microscopy, molecular modeling, the same framework, and now there are many dictionaries, all with the same syntax, that are built so that we can continue to archive data as new methods come along. We do the validation of the data, is it right or wrong? Lots of programs involved in doing that, lots of issues having to do with how you do that, and then annotations on the data, which are both, they're in some ways opinions and some ways rigorous annotations, and that's an issue. The next round of what we're doing is going to involve community annotation, which, as you know, is a very difficult and big issue in how to deal with the data, and where you draw the bright line as to what is fact and what is opinion. UCSD and SDSC is responsible for data out. For the search engines, I think we're up to our 3rd generation of search engine. We're up to our 2nd generation of website. And as new technologies come, we have to re-engineer, and all the time the data has to stay in the pipeline and never be disturbed. And there was one point where we had 3 websites that we had to be maintaining at the same time as we were going from our old system to a new system, and then the sort of intermediate test system. And it was really trying and difficult. So we were very happy when we finally got rid of the system that we started with in 1998. And then they're responsible for all aspects of data distribution. It used to be that the relationship between Rutgers and San Diego was very clear. That is, they did one thing and we did something else. But as time has gone on, data in and data out merges. And so now we have much more integrated software than we had when we began, when we really looked at it as 2 different shops. Now, because of the internet, because of video conferencing, we are doing much more integration. So this again shows people. This is the Rutgers team from a couple of years ago who are responsible for data curation. And every week we get about 150 data sets. The data sets, as I said, each data set contains coordinates, which are lots and lots of X, Y, Zs, and then about 400 or so different metadata items, and we have software that handles all of it. But in the end, these annotation staff have to really check everything and make sure that we're serving really well-curated data. The San Diego people, they don't have to stay in the stairwells. They can be outside and have parties. And so this was the San Diego group celebrating our 1st year of operation in 1999. Communication in this collaboratory that we have, VTC is really important. Electronic mail, forums, wikis. We have very clear operating procedures that we have to write and stick to. We have an internal newsletter which is very gossipy and is just among ourselves because, again, there are people here who need to know one another in order to work with one another. And so we have this internal newsletter. And then we have retreats. At the retreats, we have team building exercises, management training, technical discussions, and then time to get one another, right, to get to know one another. And at the beginning, we had to have a lot of help in terms of team building, and we had actual formal training and tree hugging exercises and stuff. We don't have to do tree hugging anymore, but we do spend a lot of time working together and trying to work out we're in the same place at the same time once a year. And this is a picture of the last retreat, which was in Temecula, which is in wine country. It's the San Diego version of wine country, so it's a little hot. But at any rate, this is the whole team for both San Diego and Rutgers. And it takes this much of a village to handle the data that we have to handle. The VTCs, we have 2 formal VTCs a week. That is, they're scheduled, and we always go to them. And then we always have the VTC on, so that anyone who wants to go and talk to San Diego just does it. And it's important to see the people that you want to fight with. And making that as a joke, but it's not a joke. I mean, that's how we have to do it. So VTCs are extremely important. Skype is okay and all that, but it's really nice to have a big room where you have a big screen and you can talk to each other. In the in this 21st century, again, there's more and more structure studies. Structure genomics really took off. Our contract was renewed. We have not had the privilege of being funded in any continuous way. It's always a hassle. And I think it's disruptive for a data resource to be reviewed this many times, but that's how it is, and we basically learned to live with it. We then decided two big things happened. There's a group that handles NMR spectral data called BMRB, and they joined the RCSB, and then the worldwide PDB was formed because we didn't want to have the situation where the structural data was being handled in different ways around the world, and we needed to make sure that we kept the archive uniform. And so we formed this organization called the Worldwide PDB. And this shows you the RCSB. That's sort of the American organization, which now consists of Rutgers, UCSD, and Wisconsin. NIST is no longer part of this organization for a variety of reasons, but there are these three universities part of RCSB. And then the worldwide PDB consists of the RCSB, the MSD project, which is at the European Bioinformatics Institute, and PDBJ, which is in Osaka. And this is a picture at our meeting that we had this summer. We did a joint booth at one of the international conferences, and there I am in the middle, and Kim Henrik is from EBI, and he handles the European data, and then Haruki Nakamura handles the Asian data. The mission of the WWTP is to maintain a single archive that's freely and openly available. And again, there are always threats to people, from commercialization and all sorts of things. And to us, keeping these data out there is really important. We have a website. And this shows now how the organization is kind of growing and who works with whom and all the intersections. And the thing we all have in common is that we are all trying to keep the data so that we keep to a set of standards that everybody will agree to. So the WWPDB is a formalization of current working practice. Right now, the members of the RCSB, PDBJ, and MSD, which is from the EBI, we signed an MOU in 2003, a Memorandum of Understanding, and it was announced in Nature. And now the BMRB has just joined us, and again, they're all the members in the worldwide PDB issue IDs that serve as distribution sites for the data. One member is the archive keeper, that is us. So only one group has write access to the archive, which is very important. Again, this tremendous fear that we had about the data getting all fragmented. I mean, for 35 years, we've managed to hold this thing together, and we didn't want to get it blown out of the water by some silliness. So there's all the format documentation's available. There are very strict rules for the redistribution of the files, but all the sites can do whatever they want in terms of making websites. So creativity is not inhibited in any way. You can do anything you want, just make sure that the data are kept uniform. But do as many different interesting things with the data as you wish. What's the future? By 2008, we estimate there'll be 60,000 structures. By 2010, we estimate 20,000 depositions a year. Right now, we have about 6,000 or 7,000. We have an equation that has worked from the beginning, so that's how we can make the estimates. The complexity of structures will increase dramatically, and new methods that we don't even know what they are will yield those new structures, so we just know that by experience. Our scientific challenges is that the number of data files will continue to increase. The information content of each data file is increasing. So little by little, we're going to go from 400. We have a potential of 3,000 metadata items that can be filled in with data. And the question is, how And the only way you could do that is by automatic capture of data from machines, and that's beginning to happen. And so that will happen. We'll have more very large complexes. There will be new methods, and structural genomics, of course, is going to yield more structures. The technical challenges is how do we represent diverse data? The X-ray crystallography, NMR, cryo-EM are all different methods, and the way in which we represent these data are different. How do we make a searchable database? It's very hard to do when you have such heterogeneous data, and that's something that we're always challenged with. And that's why the 3 sites are each trying in their own way to make better search engines. And the more people who try that, the better off we'll all be. How do we integrate with other data resources? I'm not talking today about that issue, but we do have to do data integration. And there's something like, well, there are hundreds of other data resources that have some relationship or use in some way our data. And we had to come up with methods to be able to recycle. So what happens is every week we push the data out. These other data resources come and take the data and put it and add value to it and make new annotations on the data. And now, at some point, we take that data back and we put it into our system so that people can see that, and they know for sure that it's this other data resources interpretation, but there are certain interpretations now that are being thought to be very valid and should just be in our database, and there are other things that have to be linked, and there are other, I mean, there's many different ways that we have to handle this problem. And so integration is a non-trivial thing that we are dealing with. How do we make a scalable system? If the data are increasing the way they are, we can't just hire more and more annotators to do it. We have to automate our systems, and that's a considerable effort that we are making now to do that. And how do we meet the needs of a diverse community? A structural biologist like myself understands what all this stuff means. I can go to the website and figure out what this means. But we need to get to people who do not understand molecules at all and be able to make the information intelligible. And so we've actually been spending a lot of time on outreach activities so that is happening. And we've been working actually with high school students and the like to see, what works and what doesn't work. And I'm teaching an undergraduate seminar in the spring right now, and these kids from the New Jersey high schools, I was really shocked that it used to be that nobody ever heard of molecules or proteins or anything, and they all knew how to use the PDB. They already knew I was quite shocked, and in a happy way. So I think little by little, the sort of molecular view of biology is trickling into the educational system, and that's part of what we feel we have to do. Structured genomics is the next step beyond the Human Genome Project, and it's basically to lead to an understanding of structure and function, and to obtain structural models of all proteins identified by genomics. And so this is happening now, and this puts a huge burden on us. We have to maintain many different databases having to do with structural genomics. And this is just a slide from September showing all the structures that had been determined by structural genomics as of that moment. And the guy from NIH always asks us to do this slide, and so we do it. It's fun. So who are the community? They're the depositors who have different methods, X-ray, NMR, cryo-EM. The users, specialists like structural biologists, generalists, people who are educators, students, and the lay community. And we know that we're seeing users from all those communities. So we do very active outreach. We do electronic, of course, with our website. We have meetings, we run workshops, we have publications, lots of one-on-one. There is no substitute when you're really struggling with how to make something work, to just go somewhere, visit someone, and say, how can we do this better? And then, as I said, many workshops. Issues that we have. What is the role of the centers and what should it be? This is for the members of the WWPDB. When you set standards, if you can't set standards and say, we are the PDB and we're going to tell you what to do, because nobody will accept that. So you have to figure out how to work with the community to help you figure out how to set the standards. And so a lot of our workshops, we bring the experimentalists and they critique our dictionary and say what is and is right and wrong about it. And that's how we work with the and that's what our role is to bring the people from the community in and pay them. Really, we get little grants to do this to help figure out what the next level of standards should be. Long-term preservation. How long? What are the options? Should we have these data forever? If you talk to some people, yes, but then which data should we have forever? We have all the papers from 1971 still in boxes, and we talked to Michael Lesk about what to do with this stuff, because it turns out to be a very difficult problem about what you keep and what you throw out. Stability. The strong dependence of the research community demands a more stable model. So all the interdependencies within the WWPDB, between the scientific community and WWPDB, call for a new funding model that will ensure long-term preservation and availability of the research data contained within the resources. So right now, we're on a 5-year cycle, EBI is on a 3-year cycle, PDBJ is on a 3-year cycle. The cycles are all different. If any one of us collapses, the whole show goes. So we all support one another in the funding. But this is not about cyber infrastructure. This is about funding of long-term digital collections and how you do it and how you make it stable. And it is not stable the way it is at all. And it's something that is of tremendous concern to the practitioners of well, people who are doing structural biology and people who depend on us. At any day, we have between 10 and 20,000 individual users coming to us, and every week we get tons of data downloaded by different organizations who build new databases and things. All those people would collapse, and their research would collapse if we collapsed, and yet there's no funding model that makes any sense. So this is who we are right now. The PDB is in America is operated by Rutgers and UCSD. We're part of the worldwide PDB, and we have funding from both from NSF, DOE, and 6 institutes of NIH. And we're very grateful for the funding. We just wish we didn't have to worry about it all the time, but so does everybody else. So thank you very much.
If I can ask, do you use any of the sort of standard middleware tools you use, like Globus and those?
We use at any one time we use whatever is out there, as long as it's freely available. We don't buy anything. Okay.
So what tools are you currently using?
Hibernate, MySQL for the database. The whole thing is Java-based right now.
You know if you're using SharePoint?
I don't know that. I don't know that.
You know if you're using Globus?
There was a big scene about this. I was part of NPACI, and I don't know now. I don't think so. Okay.
Are you running into any? I mean, obviously, at least if I can believe what I read in the newspapers, which is about my acquaintance with this sort of stuff. It sounds to me like there's a big problem these days of commercial people trying to patent everything.
Oh yeah.
Is that an issue for you?
Well, I was also on the National Academy Committee on Patenting of Proteins and DNA, and we said don't do it. It's not an issue.
So they haven't accepted patents yet for protein structure?
There are patents. There are patents for some proteins, but it's not a popular practice. There were many patents for DNA sequences. Oh, sorry. Sorry. Okay. There were patents, but in terms of protein structure, there's relatively few. And in this study, which was just published, we said don't do it at all. But we do have an arrangement that we're trying to make with the patent office that once the patent is issued, the coordinates have to be in the patent. So we can take it back and put it into the PDB. But it makes no sense to patent this stuff. The real challenge in drug development comes long after you find the target. It's optimizing the small molecule that you're testing as a possible drug that takes the 10 years. It's not finding the target. So it's stupid. For example, the HIV protease case, all of the drug companies knew what the structure was, and from there they competed on finding the optimal drug. So they knew the target. Yeah.
Could you, with a simple in-mid-air pie chart, give a sense of where the money has come over the last 5 or so years? Is like NSF 75%? I mean, what's the rough?
I don't actually know. We have one grant that's administered by NSF and contributed to by NIH. My guess, we're funded at about, for the RCSB is funded at about $5.5 million a year, and of that, my estimate is half of it is from NIH. That's my estimate. I have never seen the numbers, and I actually don't want to. The total funding for PDB worldwide, we estimate, my colleagues and I estimate it's about $10 million a year. We estimate that the funding for archiving represents less than 1% of the funding that goes into structural biology, determining of the structures themselves. So we estimate that there's been about $1.5 billion put into the actual determination of these structures. So there's a very interesting funding models that one could imagine in terms of, it could be like a journal model, and that could fund the whole thing. When we raised this, nobody likes that idea. I mean, the funding agents don't like the idea. The rest of, I think other people would like it because it's such a small percentage of, and it's going to go down because the cost per structure for archiving does go down because of the tools we use and all that. So if you read some of these reports that have been written by NSF on long-lived digital collections and all that, the PDB is the poster child for all of this because we've been doing this for years. And the thing that bothers me is that so far, the people who are involved in the PDB are passionately devoted to this and will go to the ends of just something like what you said, we'll go to the ends of the world to keep these data out there. But, you know, I'm no spring chicken, and none of the other people are. So at some point, we've got to have a better system than this. And I think people completely underestimate what's involved in taking the data from the experimentalist who has it in some hodgepodge way. I mean, they do their best, but they sometimes do funny things, to something that you can pick up and use. It's very difficult. So people don't really understand what's involved in doing that. Why did you ask about Globus? It was something I try to put out of my mind.
I'm trying to figure out how, you know, if our goal on campus is to figure out how to facilitate e-science.
Mm-hmm.
The question is, how much should we focus on trying to locate a set of sort of tools that are out there as standard tools for the community versus working with specific projects to just like help them do what they want
Well,
to do.
I think what our experience is that today's tool, you know, is the hot thing. Everybody says, "Oh, this is great," and then the next 2 weeks later, it's dumped, and somebody wants to do something else. And I never worried about that. I think what's most important is to focus on the application of what it is that you need the tool for, and then, because it's going to change. Whatever is good now is going to go away.
Well, okay, so I mean, what I'm trying to think, what is the role for? Presumably, I'm never going to have a huge staff, so I'm going to have just a few people to help people around campus. So what we have to do is focus on where are there. You know, where does it make sense to build expertise in a central sense that, you know, maybe there's some commonality so that when you're starting a new project, you can learn from other people. And it's, you know, I'm a computer scientist by background, so the obvious thing for me is to say, well, there must be some magic bullet that comes out of the computer science community that, you know, will solve everybody's problem.
Well, and when we have our group meetings, the people have talked to me about magic bullets all the time.
Yeah, well,
and then they change their mind.
I was going to say, it's becoming clear that's not the case. And so what we probably need is a small team of people who just sort of work as members of various projects around campus and try to...
Why don't you get a consultant staff, like if you said...
Well, that's essentially what we're looking at.
What I'm saying is that there are some really terrific projects on the campus where people have had experiences, and so you can say, you know,
right.
What you form like an organization of those projects, and then you say, what are you going to do? Like, what's the best database management system? And you can hear what people have to say with the pros and cons.
That may well be the approach.
Because one person's application is going to be completely different, I think. I don't know what you feel about that.
I agree with your points. I mean, that's sort of what I was saying. Technology is the thing that you pick and choose at the moment when they really solve the problem you have. Rather than technology, leading you to technology is the mistake.
So basically, we just, you know, we say, what is it that we want to accomplish? And then, how can we do it without spending any money?
Well, I think that there's another point of view. There's another sort of thing to think about. You mentioned that PDB is the poster child for large-scale data activities.
Yeah.
Right? Imagine what PDB might have needed 20 years ago and the kind of support and infrastructure it might have needed to think of the next meeting, the next 20 meetings, or the next 20 things like it. And are there things that you could have used in the first 5 years that now it's kind of different, right? You need a good connection and, you know, good solid power from the wall, and, you know, good tape backup that doesn't deteriorate over time. But by and large, a lot of what you do is well understood and evolves slowly, very naturally, based on the needs that you can figure out very rapidly. But what would have been useful to you in year 5?
So one of the things that we did last summer is we did a retrospective. And we actually went into those. Remember I told you there's all these paper files that we don't know what to do with, so we decided to look at them. From Brookhaven, from when the PDB was at Brookhaven, and we did an analysis of what the data distribution practices were and how they actually did that. It was very, very educational. And what it let us do when we saw that, and there were all these graphs that we drew and all that from all of that, then we were looking at another data resource that we work with. And we said, oh my God, this guy is using 1980s technology. And we didn't even realize it, but we could put his resource on our graphs in the 1980s. So I talked to him a little bit about what he was doing and why he was doing what he was doing. So you can learn, but the problem is, can we learn now from looking back then? How can we do that crystal ball that you have? And you know, you had your wish list. So